Okay, thank you. I'm really happy to be here. Thank you, DCNA, for giving me the opportunity. Next 45 minutes, I'm going to tell you everything about my work in restoring invertebrate herbivores, and more specifically, restoring Diadema antillarum, long-spine sea urchins, because most of my work for the past 5 years has been about restoring that specific herbivore. And you see the small settlers over here. For the people on Zoom, my name is Alwin Hylkema. I work from Hall Larenstein, University of Applied Science, and Wageningen University. And in this presentation, I will tell you a little bit more about how I think that invertebrate herbivores can help us restoring the resilience of Caribbean coral reefs. What options do we have to restore specifically Diadema, and what are the other options in terms of herbivores and where we are at the moment. So I'm going to start with this picture. I think we can all agree that this reef looks pretty healthy. A lot of different coral species, some branching species providing shelter for fish and other marine organisms. Maybe the fish is the only thing that could be a bit more, but unfortunately it's getting more and more difficult to make pictures like this in the Caribbean. And especially on Ceiba, I'm based on Ceiba, a lot of reefs look more like this, unfortunately. So not so many coral, also not so much three-dimensional structure, so less shelter, less productivity, and a lot of algae actually. And those algae are very problematic because they prevent corals from recruiting and recovering after other disturbances. So I made a graph with two hypothetical reefs. And the first reef is in the Pacific. And I'm not saying that all Indo-Pacific reefs look like this, but this is the coral cover. And this is from 1970 around like now. So if this reef is free of local stressors, it will of course be impacted by global stressors like climate change. And it will bounce a bit back and forth, but there will be recovery after each disturbance. Yeah, so very hypothetical. But now we have a Caribbean reef. And a lot of Caribbean reefs, after disturbances, they do not recover. There's a period of stabilization, and then there's another coral disease. And then there's stabilization, and there's a mass bleaching event. And very few examples have been recorded in the Caribbean. The only recent one I know is actually on Bonaire, after which recovery took place after a disturbance. So the problem, oh yeah, this graph is to show I'm not entirely lying. This is the Great Barrier Reef cover, Central Great Barrier Reef core cover from 1985 till 2020. And you see huge fluctuations, but after disturbances, there's also recovery. And of course, the coral diversity does not necessarily have to be the same here. Maybe these are very different species, but at least there is recovery. And in the Caribbean, we lack that altogether in many places. So the problem, or one of the big problems for that lack of recovery, is macroalgae, and to a lesser extent maybe turf algae. They are very intense competitors of corals, and they inhibit coral recruitment. So this small coral will have a very difficult time growing out of this macroalgae mat, will grow very slow and has to compete for space, for nutrients, for lights, and will likely not make it. And sometimes algae can even kill and overgrow adult corals, as you can see over here. So that's really a big problem. And what's then the difference between the Caribbean and the Indo-Pacific, why these graphs can look so different? So the Caribbean have a lower overall herbivore biomass. So we simply have less herbivores, basically. There's also higher algae growth rates in the Caribbean, potentially because we have more nutrients or more trace elements that can also limit algae growth. And very important, there's less functional redundancy in the Caribbean. So that means that there's less number of species that can take the same niche. and that's problematic. So, for example, yeah, we probably all know that the Caribbean has 2 or maybe 3, depends how you look at it, Acropora species. If you look at the Indo-Pacific, most regions have up to 60 Acropora species. So that already shows that there's much more functional redundancy. And then if the Acropora would die for some reason, in the Indo-Pacific, there's a couple other genera that are also branching and providing shelter. And in the Caribbean, those genera do not exist. Well, the same is true for herbivores. So there's less groups of herbivores in the Caribbean. And I think you already know where I'm going, but when this species, the Diadema antillarum, long-spine sea urchin, died off in the 1980s, there was no other species to take its niche. So 99% of the long-spine sea urchins died, and that resulted in a phase shift on many Caribbean coral reefs. And this by itself, the removal of the Diadema, that did not kill the coral. There's many other things that did that, like disease and bleaching. But the reduction in herbivory resulted in a reduced resilience. So the coral could not recover anymore, or the reef could not recover anymore from other disturbances. So after the 1983-1984 mass mortality, there was an increase in algae, macroalgae and turf algae, and a decrease in crustose coralline algae and corals, and also a reduction in coral recruitment, leading to this reduced resilience. The recovery of the Diadema was very slow, but in the few places that it did recover, and Harris Lessius estimated in 2016 that around 12% of the pre-die-off populations had recovered. In those few places, there was a phase shift reversal. And you can see that on this picture really well. Here in the back, we have the Diadema zone, where Diadema did recover. It has less macroalgae cover, higher coral recruit densities, and those coral recruits grew faster, survived better, and it also resulted in more coral cover. And then here in the front, we have the algae or Dictyona zone, and no Diadema. And all these metrics were reversed. So it's not surprisingly, I think, that many people think that Diadema restoration can help the reef to recover. It's one of the few things next to coral restoration and artificial reefs that we can do as intervention, doing locally. It can increase the reef resilience, and it can facilitate natural recruitment. So that's great. And we only have to do the herbivores, and then the reef can recover by itself, or it can increase the survival of coral outplants, which is of course also great. And in some places, you can even ask yourself the question, does it make sense to restore corals if the reef looks like this, if it's immediately overgrown with coral? So on Seba, we think that razor restoration has to happen before coral restoration can really take off. So the last 5 years, we worked into different approaches. We worked into optimizing some of them, and we developed new approaches to restore Diadema. These are sort of the options we have now. We can, of course, translocate them from one area to the other, but you can only do that with very high densities. So it's not really applicable on a large scale. Maybe only if a harbor is going to be dredged and there's a lot of Diadema, you could use those to restore a reef. So I will not go into detail on that one. Then we have the collection of settlers, or potentially even recruits. Grow them in tanks and then put them out on the reef later. We developed an approach called assisted natural recovery, in which we actually do not touch the Diadema. We do not necessarily even have to see the Diadema when they're small, and it still works into getting more Diadema on the reef. And we can culture them from gametes, so that is also an option. And we recently started a pilot with assisted mass settlement. So I'll also show you something about that. So I'm going to talk about the last 4 methods, basically. First, I wanted to show you like a small Diadema because they really look cool, I think, beautifully colored. They already have the spines, but many people don't know that they're bright red when they're young. They also see it moving with the tube feet there. So when we wanted to start with collecting settlers, we first wanted to compare different settlement materials, because others had already worked in this. Rolf Bach in the 80s used panels like this, like egg crate, putting it on the reef, and just going towards them and counting the number of small diadema recruits in them. So that's the only pre-die-off estimation of settlement that we have. And then Stacy Williams in Puerto Rico did a lot of work on using astroturfs mid-water to collect them. And next to that, we also compared freight rope, because it's used a lot in the shellfish culture. So we thought it would potentially work well for diadema as well. We used a dormant material, and we used bioballs. Bioballs are used in aquaculture and ponds and aquaria. They're plastic balls, around 2 centimeter diameter, and they are created to have a lot of surface area for bacteria. We thought a lot of surface area is probably also good for settlement, so we're going to try those as well. And then we actually found out that the bioballs and the dormant and the astroturf all worked well. Basically, diadema settle on anything you put in the water column, but the freight rope, for example, had to collect a lot of sediment, so that's not ideal. And the bioballs gave the most consistent results. So we continued working with the bioballs, but like dormant materials and astroturf, they can work as well. So we studied the next thing you have to determine, like, where do they actually settle, because settlement rates are not the same in every location around your island. So we're sort of lucky that we work on Seba, because Seba is pretty small, so we managed to deploy settlement collectors at 9 locations around the island. And this is basically going clockwise from the harbor, which is south. So I made the crosses green if they had a lot of settlements, so those are these 2 columns, Letter Bay and Torrance Point. Orange for intermediate, and red for not so good settlement rates. And one of the things that I think is really striking is that the windward side of the island is not so good for settlement, and the leeward side of the island is much better for settlement. And the in-between settlement happens in the north and the south, where the currents pass. And we think settlement queues might have to do something with it. It takes a little bit for the sea urchins to go through metamorphosis. And if they encounter settlement queues here, they might be swept by the current already towards these sites before they actually manage to hold on to something. And then there's potentially eddies at the back of the island. So the larvae are circulated there and they have multiple chances of settling, while in the north and the south, they're just passing it and won't come back there. So for Seba, this tells us, like, okay, if we want to collect large number of settlers, we have to go in the lee of the island. One of the other conclusions of these studies is that settlement rates were actually in the same order of magnitude as pre-die-off densities, measured by Rolf Bach in the 80s, before the die-off happened. So that was really surprising. Apparently the number of larvae was not really limiting natural recovery, but other things probably do. Settlement over the year was still a little bit lower, and I already told, like, bioballs were the most effective, consistent in collecting settlers. So what do you do if you have the small settlers? We set up like a small nursery. As you can see, it's pretty low tech. It's all standalone recirculation system with air-driven pump filters. We use LED lights to get some algae in there for the settlers to eat, and they feed on the biofilm for the first couple of weeks, and then we feed macroalgae to the juveniles. Well, we use 12 hours light and 12 hours dark, so this is a very simple approach to grow the settlers that can be done on basically every location. The size of the settlers is 0.4 to 2 millimeters large, and after 6 months you have juveniles of 1 to 3 centimeters, and then after around 3 centimeters we think they're ready to put back on the reef. We have in that period, so growing them from settlers to like juveniles, we have 30 till 70% survival, and that survival depends on the size that we get them. The bigger the better. The stocking densities, if we put too many in a tank, the survival goes down. And micropredators. especially with feeding them algae, we introduce small crabs, fireworms, and they really can decimate especially small settlers. So we, once a week, if they get a little bit older, we really go through the whole tank, turn everything upside down to search for pleasure, to look for small predators. Then, of course, after they're big enough, we have to restock them, and that process is not different than culturing from gametes. So I will cover that later in the presentation, because of course we still have to put them on the reef. So this method, comparing it with the other methods we have, is really low tech, low cost. Probably you have to invest like $1,000 in equipment, and if you have a room somewhere, you do not have to climate control it or something, it can work. But first you have to do quite some work, because you have to identify the settlement hotspots and the settlement season, which for the northeastern Caribbean is really in summer. So they spawn in spring, and then settlement is in summer. The problem with this method, or a disadvantage, is that it's dependent on the reproductive season, and that shifts throughout the year, so it's not always exactly the same. So you need to have your bioballs in the water for a long time to make sure you don't miss the settlement peak. And you're dependent on source populations, of course, and not every island in the Caribbean or every area in the Caribbean gets as good settlement as we do. And at the moment we're doing a study in 15 different territories across the Caribbean, and we're doing this for 4 or 5 months now, and we plan to continue for a year with a lot of different partners. And that will give us a better idea how settlement is distributed throughout the Caribbean, but we can already be fairly certain that in some places this method won't work, because there's not enough settlers or larvae available. At a certain moment, we were seeing diadema everywhere. Well, when you work a lot of them, you see them everywhere anyway. Like if you have been through those containers with the bioballs searching for the small spots. I was taking a shower that afternoon, and I was seeing small diadema on the sides of the tiles everywhere. So you see them everywhere anyway. But I was also seeing them on nursery trees, for example, and on buoys in the water. And those were much bigger. So at a certain moment I thought, are we not wasting our time looking for these tiny settlers? Should we not let them grow a little bit, and then we can potentially put them on the reef as recruits, and we don't have to grow them inside. You cannot do that on the bioballs, because the bioballs have spaces of 2 millimeters. So within a month they grow out of the bioballs and would fall off them. So that wouldn't work. So we designed a new bioball, which is much bigger. It's like 12 centimeters in diameter, and it still has the same 2 millimeter plates in the middle, but then if you go out, every other plate is removed, so you end up with a growing space of 2 centimeters. So potentially diadema up to 2 centimeters can grow in that. And we did an experiment with that 2 years ago, and it worked. As you can see, we had quite some large juveniles that we could collect, but not in the same numbers as we expected to, because we also took out bioballs after a month and after 2 months, and we had quite some settlement, but not so many recruits. So something happened, they fell off, or what we think happened is that they were eaten by crabs, because those small, narrow spaces were also excellent for trap recruitment. That always happens if you try to facilitate one species, you accidentally always seem to facilitate its predator or something. Very funny, all these bioballs were kept suspended in the water column by small buoys, which you see over here. And then we had a lot of diadema on the buoys, so you can see them here, and here in the bottom, and sometimes we had 5 or 6 small diadema on these buoys. So in total we had, I think, 12 after 4 months on the bioballs, and we had like more than 50 on the buoys, which were only meant to keep the bioballs in suspension. So the year after we did another experiment. Here you also see quite a few. It was last year we did another experiment with buoys and then drilled holes in them so the crabs would not no plates but holes. And that worked too, but the settlement was not so great last year. So we cannot really prove yet that it works, but I think there's a method there to potentially even skip this outgrowing phase and be able to transfer them straight onto the reef. But this is still a work in progress. Then the second approach that we developed is the assisted natural recovery approach. So one of the things I thought, like, okay, larvae are not limiting, right? There's enough settlement. Settlement is comparable to pre-die-off settlement in Curacao. So something else is happening, probably in the early life phases, because we don't see the recruits on the reef. So we have the larvae, but we don't see the recruits. Somewhere in between it goes wrong. And I thought, what if we put those bioballs as a string? So these are the bioballs, then we make a string of them, 50 bioballs, and we attach them directly on the reef. So they have a settlement substrate, and they are also a little bit protected in that very vulnerable first life stage. And then we don't remove the bioballs, we just leave them there. So they outgrow them and then fall on the reef, and maybe we can increase Diadema recruitment on that patch reef there. So we, to find out what was happening, we also placed proxy streamers, extra streamers next to the reef, same conditions, and we would replace them monthly to get an idea of what kind of settlement was happening. So these are the results of the proxy streamers. And it basically shows that we did this in the right time of the year, right? Like right before the settlement peak, we put them in, we had some settlement, even better settlement, and then that was the end of the peak, and we still had a little bit of settlement afterwards. So that was the first, and you could actually see the small settlers sitting on the bioballs on the reef. So that was pretty cool. And then we compared, of course, every month we counted the number of recruits on the reef. And the blue line, dash line, is the reefs with the streamers, and the red line is of the control. And you see that both initially increased. So that's just because it was within the settlement season. But there were more recruits, and they also increased for a longer period of time on the streamer reef. So the results, or the method worked, and we published it, but we think some optimization is still necessary because the numbers were not big enough to result in permanent population establishment. So after a while, after a couple months, the recruits were still eaten, probably. When we came back after a couple months, then there was no difference anymore. So some conclusions here. I think this is a really low-cost method and also low-tech. Almost no effort necessary. Investment of equipment is also really low. Just a couple bioballs, a couple tanks, $300, I estimate. And this has a lot of potential for large-scale application, but needs to be optimized. And I think also try it in different locations, because it might also be that Seba just has a high density of predators preying on these recruits. Maybe this goes much better in other places. And of course, some time is necessary before you can start this, because you first have to know where your settlement hotspots are. If you just attach the streamers on any reef and any time of the year, then the chance of failure is pretty big, I would say. And even if you know that, the results will be uncertain. You cannot really observe the process, and there's yearly and monthly fluctuations in settlement. Another disadvantage is that if you're too early, then the bioballs get overgrown with more longer turf algae, and they do not seem to work as well as a settlement queue anymore, or there might be too many predators in there. Because we tried with older bioballs, with an older biofilm of like 3 months, and then there was almost no settlement happening on those. So from assisted natural recovery, we moved towards culture from gametes. And Diadema always have been known as a very difficult species to culture. So we didn't want to make it too difficult in the beginning, and we actually thought we would not be able to culture large amounts. So we just wanted to culture them to do some settlement experiments to find out what the queue was that makes them settle on the reef. So the purpose was to culture some competent larvae, find out the queue, but then actually the method worked better than we expected, and we now think that this is potentially the best method for upscaling to produce large numbers. What we did, we compared a couple different methods. We compared the chrysal vessel with a couple other things, and also with the shaker table or shaker bottle method. So we use an orbital shaker table that sort of keeps the larvae in suspension, because that is the trick. If you don't do anything, they will sink to the bottom and at a certain moment die. So you need something to keep them in motion. We collect adults on the reef, and we give them a warm water bath. It sounds much nicer than a heat shock, I think. That's the alternative. If it's summer, we first have to cool them down for only a week. So it's 30 degrees on in the water now. So we take them out, cool them down to 25 degrees, and then within a week we can spawn them with a warm water bath. And we have pretty high success with that. We use then the orbital shaker table with 1 liter bottles to keep them in suspension, and we feed them with Rhodomonas microalgae, which is very nutritious microalgae. After 25 to 60 days, we have competent larvae, and this has been demonstrated, this method, in the Netherlands, where we first developed it. We transferred it to Seba, where we use it now, and also to the States, where we work together with a partner that also tested this method, and it works there well. What was really great about this method is that it's very easy to have a large number of replicates. Because just a couple bottles on each table, a couple tables, and you easily end up with 20 replicates. So we could figure out all the culture parameters, like what do we have to feed them, the feeding density, the temperatures. So that's definitely the advantage of this method. And here you see the different stages, like basically from an egg till like a larvae, till a competent larvae that has a rudiment. You see the tube feet sticking out here, and here it's a settled urchin. To make this step, this go through the metamorphosis, the larvae needs a certain cue to go through settlement, and that's how we started this culture. Actually, this is what we wanted to find out. So we did 10 larvae in each petri dish, and the petri dish had different treatments. So we compared crystals coralline algae with a whole bunch of different macroalgae, Herbivora, Laurentia, Pedina, Hadimeda, Dictyoda, and Sargassum, all common species on the reef or even in the water column, if talking about Sargassum. And of course we had a control without algae. And then actually settlement was much better for most algae than we expected. What we really expected is that CCA would do really well, and that the other algae would not do so well, and that we actually thought, that was our hypothesis, that the change on the reef that we saw for the last couple of decades also resulted in less diadema settlement. We cannot really conclude that from these data. We actually see that they settled pretty good on most of the common species. They still like CCA. And we did another experiment with a different culture run, and then compared different biofilms, and there seemed to be sort of an attraction, or biofilms of 2 to 4 weeks seem to be like a really good settlement cue. And then if the biofilm gets older, it seems to be a little bit less attractive, but not clear enough to show statistically. So this also corresponds with the bioballs in the water, right? Like we leave them there for a month, so the biofilm will be on average 2 weeks old when they settle, and that's actually pretty good. So that's probably why that works. And we also included CCA here as a control, and this run had overall better settlement. So it's good that we took the CCA as a control. Otherwise, we would think that the biofilm would be better than the algae. So of course, we still have to grow them, but that stage is not so different. It's sort of similar as collecting them from settlers. But like around 2 years ago, we thought, okay, we can now grow them, we can settle them, and we can grow them out. What are existing bottlenecks in cultivation? It's for sure not the amount of gametes that one female produces, because one female can produce 5 million eggs, or up to 15 million actually. So that's not an issue, and males produce even more sperm, so that's not limiting at all. But our first bottleneck was in the culture vessel. Like on one shaker table, we can keep around 1,500 larvae in culture. Of course, we can put a second and a third, shaker table there and have more people caring for them. But it's not like, yeah, you would still end up with only like a couple thousand larvae. Not enough if you realize that only 10% of the settlers survive the first month, and not really shown here, but not all larvae make it to competency either. So you have like just quick steps in succession that not all make it to competency, and then the one that go through metamorphosis, not all of them survive. So we end up with around 150 juveniles, which is still nice, but then you put them on the reef, and then there's a very low survival, and the number that you are able to keep there, that's not enough for permanent population establishment. So these are sort of the bottlenecks that we wanted to tackle, because that is what is necessary to use this approach for really restoration. So we started the Diadema 2 project this January, actually, which is really focused on targeting these bottlenecks. And of course, we start here and work our way this way. So upscaling the culture, we now use conical tanks that you see here. They are 150 liters each, and we compare upwelling and airlift methods to keep the larvae in suspension, and the results are actually much better than we expected, especially as we're only using it for a little bit. So these tanks, compared to the shaker tables, can harbor many more larvae, 300,000 per tank, compared to the 1,500 per shaker table. And in our first run, which is also our last run with this vessel, we had 10,000 settlers already, and we didn't run at full capacity. We didn't have 300,000 larvae per tank. We only had 20,000 larvae per tank. So I think these results hold really good promise, and especially because there's much that can still be optimized, like airflow rates and feeding, etc. So some conclusions before I move into the restocking part with this method is that, again, you need around 6 months to culture the juveniles. The survival of settlers is pretty low at the moment, so that's one of the bottlenecks we hope to target. Of course, this asks for much higher investment in expertise and resources. So you need at least 10,000. If you already have like sort of a lab space, you need to be climate controlled. But it has a lot of potential, I think, for large-scale application. The numbers are just much more what we need, actually. So we're working on resolving these bottlenecks, and then I think we're ready to transfer the technology so others can start using this as well. The nice thing, of course, and the big advantage is we're independent of natural settlement. We can culture them year-round. Even in years of not so good natural settlement, we can still produce a lot of juveniles. So then we have the restocking, and that is the same for if you collect settlers or if you culture them from gametes. If you keep the urchins in the lab for a certain period, they change. They change in morphology, or they develop different. They develop different in morphology, physiology, and in behavior. And that is problematic because these are the causes for this low retention, we think. If we put them on the reef, they're basically not ready. They're not field hardened. Some things have to be done there. This is images from one of our last restockings where we restocked around 200 urchins on a reef. And we on purpose chose a shallow reef, because we thought queen triggerfish, they were really problematic in the past. They don't live as shallow, live a little bit deeper, so we might be able to out-shallow them, sort of. That didn't really work, because the hogfish, Spanish hogfish, are as good as eating juvenile Diadema, as you can see here. So it took only, I don't even want to show you the retention graph, because it's terrible, it makes me cry. Yeah. And then this is from an earlier attempt on, this was not a restoration attempt, this was really a research attempt. We restocked them on larger artificial reef structures. The structures are called more reefs, and we developed them in another project. And constantly the Diadema were disappearing from them. First after 2 weeks, then after 2 days, and then after 2 hours. They were gone if we put them there, and we didn't know who was responsible for that. Of course, we had ideas. But we put cameras on there and designed an experiment, and then we found out that actually the triggerfish, that really learned quickly to eat them. And they're actually smarter than we think, because we also did it with smaller urchins, and they were not able to get to them because the Diadema could shelter really well in the mooring shelters. But then they came back around sunset. So the first day there was triggerfish activity the whole day, and they were not successful. But then the second, third, and fourth day, you saw them shifting towards this short period before sunset, and then the short period, sunset and sunrise. And they just knew that the Diadema were coming out either to feed or to look for a better spot, because they had been attacked during the day, not eaten, but then they want to move, and then they were probably still eaten by the queen triggerfish. So what's so different between lab-reared and wild urchins? Okay, who thinks that this is the lab urchin? Who thinks this is the wild urchin? So it's, okay, hands up for lab, and hands up for wild. Yeah, so this is the wild urchin. This is the lab urchin. The lab urchin has longer spines because they never get broken in the smooth tanks. And the spines are also thinner than the wild ones, and a bit more brittle as well. So that's already in the morphology. And the physiology is different. If you restock these guys, and we did that on a very shallow reef where there was a little bit of swell, and there was some natural recruits there. And you put them on the reef and you see them like, they're really struggling, even holding on, and their spines, they go like this, you know, they cannot really defend them, so they cannot even get them upright. And there's a natural recruit next to it. It's like very difficult for fish to get near to that. And then their behavior is different, and that is shown in an experiment that we did here. We had 3 groups actually, and this is the positive part, because it shows that we can easily turn this around again. This is reversible. We had 3 groups. This is the cultured group, so they're coming straight out of the lab. That's the green bars. And then we have the red. Those are booster ball urchins. I will explain in a bit. And then blue is the wild ones. And you see that the wild and the booster, they behave exactly the same, while the lab ones are really different in their behavior. So this was simply activity during the day. And a Diadema is supposed to shelter during the day because it's very vulnerable then, and all its predators are active. And obviously the cultured ones didn't shelter. And the wild ones behaved as expected. They were sheltering during the day and feeding during the night. And the booster urchins, those were actually urchins from the lab that we put on these big bioballs, because they didn't work for the recruits because there was too many crabs, but we thought we can use them as a habitat for us to put like recruits on. So we kept them there for only 6 weeks. We lost a few, but not that many, still enough to run this experiment. And then you see that only 6 weeks is enough. So they came from the same lab, same group basically. We split it in 2, kept half in the lab and kept half on the booster balls for 6 weeks. And they behave exactly like the natural ones. They feed during the night, shelter during the day, and also their morphology was totally comparable with the wild ones. So this shows we need a period of acclimatization before we put them on the reef, and then we probably solve all these problems at once. So I mean that is, I think, very, very hopeful. Then the third, this is the fourth or the fifth option that we have, and we just started with this. This is something that I wanted to do since those papers came up from the Australian researchers like Peter Harrison and others. They did this with coral larvae, like having the larvae and putting them under a fine mesh net and making them settle on the reef themselves. So assisted mesh settlement. But we never had the number of larvae to do it, because running a shaker table is a lot of work, and then you have maybe 700 competent larvae, and I mean, those are so precious, you're not just dumping them on the reef, you want to make sure that they do well. But now with the conical tanks, we actually had the numbers. So we did this 2 weeks ago. This is 1 by 1 meter, and we put like 10,000 larvae underneath in this small area. And I don't know the results yet, of course. We have to come back in like 2 weeks and 6 weeks to see if this works. It's just a pilot. But if we have some indication that it might work, then this is really something I want to explore further, because this would take out the whole difficulty of like settling them, growing them, and also readapting them, acclimatizing them to the world, because that will all be done by nature. And of course, we'll lose a lot of larvae by doing that. But if we have enough, then who cares, right? This might be much more efficient. And the advantage of culturing. In 2022, we had a Diadema die-off again. So in a lot of places in the Caribbean, Diadema died, sometimes relatively small percentage. On Sable and a couple of reefs, 99% died. This made me really think like, okay, Diadema might be naturally the most abundant grazer, so they're also really good at it. Remove all the macroalgae, but to make the system dependent again on this one species is maybe not the best choice. Because if we then have a die-off, suddenly all the Diadema are gone again. And actually restoring them in large number might actually increase the chance that such a die-off happens again. I want to branch out and look at different grazers, because Diadema are obviously not the only invertebrate grazer on the reef. And then we do not necessarily have to be dependent on one species. So some candidates that other researchers already did some work with, for some of them at least, the Caribbean king crab, the West Indian sea egg, Trypnosis ventricosus, seems to be a very suitable candidate. You see them here, the sea eggs, sea apples. And the West Indian topshell, which is more sort of just subtidal species, very shallow, but also a herbivore. So we thought that snail might also be suitable. The Reef Grazer project that will start in September, we're going to look at these different species, their effects on the reef, because for some species that has not been studied well enough, we of course want to know if they eat the algae, don't eat the corals, what sort of densities we're looking at. We also want to know their effect on each other, especially working with crabs. They're omnivores, of course. They might have an effect on each other as well, so we want to study that. And all of these species are actually edible and actively fished in some parts of the Caribbean. So we also want to study if we can culture them for a dual purpose, like for restoration and for fisheries, and if that can be combined. The fisheries would be, or fishers would be interested, and of course how we can co-manage that in a way that we're not putting them out for restoration and immediately fished away. That is a challenge. So this project will start in September. So the coming 2 years, we hope to get a lot of information about these other grazers. We couldn't wait for that. We already did a very small pilot. So here you see us working with a Reef again that was caged, and we put 3 Trypnosis on it. Within 2 weeks, you already saw that they sort of cleaned all the algae. We did the same for Diadema, sort of a control. Of course, we had real control and grazed, and we also put the whelks on there, the top shells, in West Indian top shells. The whelks did not so well, and I cannot really tell if they didn't like the 15 meters, which is a lot deeper than they usually occur. They survived, but they were not actively eating the algae, or at least not having an effect on the algae. The algae were actually more abundant after the experiment than before we started, but too early to really draw firm conclusions from that. Here you see us restoring some of the whelks. So I summarized, because a lot of you are looking into restoration options. What can be implemented now in terms of grazer restoration? And I think collecting Diadema settlers or potentially recruits, that's something that everyone can start with now if you didn't start already, if you want to do that. And first step is to determine the settlement hotspots, because if you don't know those, you might spend a lot of energy getting only a few adults. And then if you know your settlement hotspots, you can put a lot of lines and a lot of viables in those locations and potentially collect a lot of them. What can be implemented in 1 to 2 years? I honestly believe that the large-scale Diadema culture is not so far off in being able to be transferred to others, and we solve the main bottlenecks, and we can go from there. So I think 1 or 2 years is realistic to start that up in different places. not for research, but really for restoration. And then in 2 to 4 years, I think we can also look at the culture and restocking of other herbivores. But maybe even sooner, because I know other groups also work on this already. So our research priorities for Diadema is the settlement and the post-settlement survival, especially at the moment, and the restocking, because that also is not, the retention is not good enough. And then for the other invertebrates, we'll look at effects on the reef, effect on each other, and then next step, when we have selected suitable candidates, we'll work on the culture and the restocking again. And like I said, I'm based on SEBA, so we do this with a small team, work from the SEBA Research Center in close collaboration with the SEBA Conservation Foundation. This is the controlled lab, but we also have a large wet lab space to do all these experiments, and we're setting up mesocosms at the moment to do some behavioral experiments. So I think we're up to the task. So I want to thank all the partners that make this work possible, that we collaborate with, the colleagues, students and volunteers that help me collecting the data. It's by no means all my results, but we did this together. And I'm very open to collaboration with other islands, and we actually, we work together already with quite some of the Dutch islands. So thanks for your attention, and I'm open for questions.
We love your project, and it's indeed great to see that already you're working together with other Dutch islands. Because of the time, we're going to do 2 questions. So one from people in the audience and one from Zoom. And then I would suggest to do other questions during that. So. Yeah, that's all. Like I'm first.
It's 2 things. One thing is that you consider Invertebrate Laguna, because we already know how to breed them quite easily, and it's a very good species. So,
which species are you referring to?
The Invertebrate Laguna.
Oh, yeah. So those are rock boring urchins.
Yeah.
Yes, I did consider those. They're easy to culture, but their effects on the reef are not so great. They really bore in the substrate, so they're even worse bioeroders than Diadema, for example. And they create this sort of, they eat very selectively. So what you often see is that you get like a canopy of macroalgae, and then they have their burrow there, and they eat on the turf algae there. But they keep the canopy there because it protects the burrow. So no, I don't think those are suitable for restoration.
And one other. I'm cheating, sorry. No, because sargassum has such a high settlement rate, did you ever consider using sargassum? The problem also is an advantage, because I think there's less predatory fish hanging around in sargassum. Obviously, they're already, when they're on loose, it's older, but maybe help them grow a little bit further and also disproduce. It's a weird way of thinking, but maybe.
Yeah. So the question is what do we do with sargassum because it has such a high settlement percentage? We think it might potentially be a sink for Diadema larvae because they settle in the sargassum and then wash ashore, and then of course they don't really end on the reef. But I think we did some depth experiments where they settle most, and the Diadema larvae seem to be more mid-water and not really in the top surface layer. So I think in nature they wouldn't really encounter each other so much. So I think
that's why I mean maybe you can use it as an advantage, because sargassum needs to be cleaned up anyways, and if there's a higher survival because there's no fish predatory, maybe it's a win-win.
I don't really get it. Like I have to put the Diadema in the sargassum then, or because they don't encounter they don't encounter each other naturally. I think we also
but if it works better, I mean it's already not naturally what you're trying to do, so maybe 1 + 1 is 2. It's worth testing.
Okay, we talk about it later. What you want to test, because I'm still not in time.
Sorry, maybe this is one of my weird ideas.
We have a lot of questions here, and I'll ask them to each item later. One question from Lotte. She's wondering how genetic diversity is maintained in cultural environment from sea urchins in the lab. And could this also form a genetic bond?
Yeah, so of course, a very relevant question. So far, we have been really working on the research, getting the sort of culture methods worked out, the techniques worked out to do this. But then if we want to do this on a large scale and put them out on a large scale, then this is something we have to consider. And of course, we're not going to take one group of broodstock animals and then use them for multiple spawns. So one of the things we can mitigate this is always take multiple animals, so they're not all from the same adults, and also for every run take different animals. But of course, there are different approaches. I think the standard things that apply for culturing, anything for restoration, apply here as well. And it's definitely something to take care of. Okay.
Thanks so much.